So I think maybe I can start. So today, unfortunately, unlike Ron mentioned, I'm not going to really do an introduction. I'm mostly going to talk about a system we use where we study organoids and some ideas or novel ideas of how to look at single cell data to extract gene expression dynamics. And hopefully, if anyone has some thoughts about this recent directions or comments, we're currently developing this sort of way of looking at data. So I really welcome feedback. So very quickly, I'll introduce sort of like the overarching theme is how to achieve tissue engineering. So what I'm showing here is a depiction of the liver and its functional parts. So this is a liver lobule, which is the functional nerve within the liver. And to approach that, the lab has been developing a system to engineer liver-like organoids from iPS cells. So very broadly, we have this system where we use lentivirus integration to overexpress a transcription factor GATA6 into iPS cells. And what we saw after some evaluation was that this served as a cell fate switch. What I mean by that is that the iPS cells that receive a high copy number of this virus and have a high dosage of high GATA6, they follow a mesoderm to endoderm trajectory. The ones with medium levels can go somewhat either way, depending on their neighbors. And the ones with low GATA6 expression or no GATA6 continue to ectoderm, which is the default path of iPS cells in culture. Now, very interestingly, if you continue to grow these cells, what we see is that the endoderm cells will mostly become or follow a fate towards hepatocytes. The mesoderm cells will preferentially become endothelial cells, but we have also observed evidence of hematopoiesis occurring in our organoids. And then the ectoderm cells will continue to go into neurons, basically. So we're trying to understand how this system works and how it develops. For that, you see me? Okay. So since then, this was work that was published before. And since then, we have moved the system to 3D because it's more interesting, we believe, but also we believe it better reflects development as it occurs naturally. So these are some examples of confocal microscopy of our 2D and 3D organoids. So in this box, somewhat curious to see that there's an accelerated development in 3D organoids as depicted by the abundance of red, which is an endoderm marker in our 3D organoids. And also, if we jump to day 8, you can see that we have hepatocytes marked by HNF4 alpha and CD34 is marking an endothelial cells. This is a good reflection of our organoids. We have a lot of hepatocytes and they're an interconnected network of endothelial cells. Now, we really want to understand how these cells transition molecularly from iPS cells to these very different, how do they follow these developmental trajectories? So to address that question, we did a time course experiment on single cell data. So this is data. The different colors here, well, every dot represents a cell, and the different colors represent the days in which we took our samples. So we have day 0 represents the iPS cells, then we have day 3, 5, and 8. This is an early assessment. And using, oh, I'm sorry. So, well, okay, missing a slide. But anyway, so we want to see how these cells develop. What I'm missing here is a slide showing markers in which I can show that these cells, this group over here corresponds to iPS cells. And then we have 2 developmental trajectories, one along this line that corresponds to the mesoderm. And then this path represents the endoderm. So we can identify iPS cells, endoderm, and then more mature hepatocytes, again, using markers. And we want to understand how that happens. So this is the setup. And this is where we're trying to implement these new ideas of dynamics, analysis of dynamics over time. And the way I do that is basically if I take these cells that corresponds to the endoderm path, I can, using existing methods, we can organize them in what we call pseudotime. So basically, if I tell a particular algorithm that this set of cells are the starting point, we know these are the iPS cells, we can sort the cells based on their relative differences in their transcription patterns in pseudotime. So it basically just goes from 0 to 1. It represents beginning and end of the experiment. And we can see that the cells are sorted. The pseudotime corresponds to the real time as based on the timestamps. And now what we can do with this analysis is that we can look at our favorite transcription factors, for example, and look at their behavior over time. Now, as most of you know, single cell data is pretty sparse and very noisy, and this is a good representation of it. So we've been looking at ways, and this is maybe some of the things will be fun to discuss, about how to smoothen this data. Because there's multiple implementations and different ways to smoothen it. But in this case, we use a very simple sliding window average technique. It's very naive. And basically what this represents is that in the x-axis, we have pseudotime. And what we can see is, this moves on, that as expected, but as we can see, the HNF4 alpha, which is a transcription factor associated to development of hepatocytes, is non-expressed at the IPS stage. Then it becomes production in the endoderm phase, and then it becomes established at high levels in hepatocytes. So now the next question is, or the question we wanted to ask, are there other genes with similar associated dynamics that can be part of developmental circuits? Now, this is a hard problem. So to those who have some experience with time course data, the statistics are really complicated and they don't really scale very well. So when thinking about single cell data, where we usually have high dimensional data, this is not ideal. Even if we subset the data to include only transcription factors, in our data set, we still have about 900. So there aren't that many good methods, or statistical methods, or classical statistical methods to address this issue. And because of that lack, I turned to sort of novel methods in machine learning. So the implementation I'm using is algorithm that is mostly used to track stocks in stocks markets. And if you think about it, conceptually, we're pretty much asking the same question, trying to find genes with similar dynamics over time. It's very similar to ask to try to find different set of stocks that behave similarly over time. And so we can, again, use machine learning. This is an unsupervised clustering algorithm. And these are some of the results we get when we compare the transcription factor dynamics. So this is a heat map of distances. So in this case, what we show is the stronger the blue, the stronger the association between the dynamics over time of transcription factors. And something that is clear is that there's some grouping, there's some clustering. Now, in more detail, what that means is that we can evaluate our favorite transcription factor, HNF4 alpha that I just described, and we can ask, what are the most closely associated gene dynamics in our data set? And basically, we can find genes with similar expression dynamics. And something that was very nice is that we are pulling out clusters that have been reported previously in the literature, which makes me think that the analysis is working and we're getting meaningful data out of these expression dynamics. So most of what I mentioned so far is focusing on the endoderm path. Now, one of the things we really want to do in the lab is to further develop the organoids by using genome engineering. And one of the things we really like to achieve is to have specific changes to engineer changes that are lineage specific. And we can use this type of analysis to look for gene sensors or candidates to do that. And this will be one example, right? We can evaluate the expression dynamics of this set of transcription factors as you follow the endoderm path and also as you follow the mesoderm path. And this leads to candidates, genes that have lineage-specific expression, and therefore will be good sensors to produce lineage-specific circuits. Now, something else we can do, and also for further validation, is we can look at the opposite trend of decreasing expression in time. And the control here is OC4, which in the databases is referred to as POU5F1, which we know is high in iPS cells, and the expression goes down as the cells differentiate. And again, we can ask for closely associated genes, and most strongly associated gene is SOX2, which again is consistent with what we've seen in the literature and what we know from differentiation by most other means. So the idea is working, the clustering, the machine learning clustering is working and is providing a good representation of associated dynamics in time for this single cell data at high dimension. Now, just for fun, some idea that we've been considering with studying in the lab is, would we use this to learn gene regulatory networks? And would it provide some different view if we use this dynamics mindset? And one way to think about it that we're considering is, let's assume that we turn the time axis around. So if we flip time around, in this case, we would basically be looking at dynamics in de-differentiation. So going from differentiated cells to iPS. And the reason to do that is because in iPS cells, the gene regulatory networks are fairly well established. So we could have a gene network control that we know we have to aim or develop algorithms so that they can reconstruct iPS gene regulatory networks. If we could show that, then we can also go forward in time and use that to learn how the new regulatory networks are established as the cells traverse different trajectories in time. And I think that would be something that would be probably of interest to develop, and it will help us learn more about development at a single cell and genetic level. So with that, I just want to acknowledge some of the people that have been involved in this organoid work and the sequencing efforts. And if someone has any questions or comments about this sort of set of ideas of looking at dynamics expression, I'm really interested in hearing your thoughts.
All right, great. Thank you, Jose. I don't know who's actually hosting this, but we really be great to get some questions, comments from people.
And if people are interested in participating in developing this, we're somewhat early stages and we welcome a lot of suggestions.
I have a question. What's the meaning of that quasi-time?
That's a good question, a really hard one. So it's...
Because that it will matter how you average those points, I assume, if it's not equal sampling, or it depends on the meaning of it.
Correct. So yes, basically the mapping distance between pseudo time and real time is going to be a strong limitation. It could be a strong limitation in analysis. So in particular for our data set, I'll go back and show you. If you look here, we actually have a fairly incomplete sampling. So at day 3, we were not very successful at dissociating the organoids. So we're kind of missing a good chunk, or we don't have a good representation at this time point. However, I do think that what we see across time, it is reflective of the dynamics of at least the subset of transcription factors we're evaluating. So unless there's a significant spike, like it goes up and down or something like that, and we missed it, that could be a problem. But maybe what I'm trying to say is, it feels like, at least in this example, the analysis was robust enough to provide, data that is consistent with controls.
So, Jose, but if you go back to the clusters that were collected at the different days, can those serve as anchor points so that you can take kind of the middle of each day and that represents an anchor? I mean, so you can, pseudo time may be stretched or shrunk in between those median or median centers, but in between the centers it should be the same.
Yeah, I guess you could restrict the range in pseudo time by the timestamps, right?
Yeah.
So you can say day 0 corresponds to the first. It can only go up to like the first 25%. So yeah, maybe that would be a way. to determine if you have how big of the gaps, or to show more clearly show where the gaps in sampling may occur.
I have a suggestion and a question. My suggestion is, I'm not sure which clustering algorithm you used, but perhaps you could try even a simpler one that's not so machine learning based, but like Coxon was pretty popular not too long ago, where you're looking at correlations of correlations, and since you have different cell types, you could be asking and do the correlations between the different gene expression profiles over time change for your different cell types, which might be an easier algorithm than setting up something like a neural network. My question then is, in terms of the clustering algorithm that you're already using, have you noticed that biological replicates change things much, or done any kind of error on, you know, which genes cluster based on two different organoids but the same days tested?
We have not tested. We only have this one data set so far. And so that's to the second question. To the first one, if I understood correctly, or maybe let me do the clarification. One of the reasons to use machine learning is because I want to study this as a time course. I think the most common strategy that I'm aware of is that people would evaluate gene expression levels by subsetting this into different populations, right? You can ask which genes are differentially expressed at day 0 versus day 3 versus day 5. Or if you do clustering and identify the specific cell populations, you can ask for changes across. But it's somewhat of a different way to think when you want to keep the time axis. So you want to look across the whole time course. And this one, I was thinking about it. It has inherent, there's an assumption here that may not be correct. Because so if I want to look at time course data, you assume the difference would be that you assume there is codependence between the time points, right? And that each time point is correlated to the next. And this somewhat separated for a particular type of statistics. So in this case, this is why it doesn't scale well. And correlations, I think, are not the best ones to use. Now, in the real data, our cells are somewhat independent, right? So I'm sorting them out in this pseudo time axis and assigning dependency. I'm saying that they provide a good representation of what would happen in one cell across time as it differentiates. So I'm still trying to figure out, and we're discussing here, what are the implications of this lack of dependency. But at least we're somewhat encouraged by the fact that we can reconstruct existing clusters, and that some of the patterns we see are consistent with known extensions or known dynamic changes in a specific set of transcription factors.
What about biological replicates? Do you pull out the same ones if you do this, if you have two data sets?
I would like to think so, but we don't, again, we just have this one data set so far. We will have to produce more and see how they map. But my expectation would be that it should be similar. I mean, at least not by single cell data, but we have seen that the organoid development is fairly robust. If anything, we have problems changing properties of these organoids. So my expectation is if we do it again, we should see something very similar.